Hello, and welcome to our webinar, Protecting Your Vision, New Tools and Discoveries in Glaucoma Care. My name is Cynthia Steele, and I am the Chief Scientific Officer here at Glaucoma Research Foundation. Now, it is my very great pleasure to introduce Dr. Brad Fortune. Dr. Fortune is a senior scientist and principal investigator in the Discoveries and Site Research Laboratories at the Devers Eye Institute in beautiful Portland, Oregon. It's part of the Legacy Research Institute, where Dr. Fortune holds the Van Buskirk Chair for Ophthalmic Research. Dr. Fortune earned his bachelor's in biology from Cornell University, his optometry degree from SUNY College of Optometry. He completed a residency at the San Francisco VA Medical Center and received his PhD from UC Berkeley's School of Optometry. Dr. Fortune is widely recognized for his work in retinal and optic nerve physiology, with a focus on advanced methods to assess visual function and imaging of the retina. His translational research bridges laboratory discovery and clinical care, aiming to improve detection and management of glaucoma and other eye diseases, as well as explore new possibilities for vision restoration. We are here at GRF, we are so lucky to have Dr. Fortune as well serve on our Schaefer Grant Advisory Committee. So please join me in welcoming Dr. Brad Fortune.
Hello, and thank you very much, Cynthia, for the invitation to be with you today. Our laboratory receives fees through some consulting I do for the companies listed on the screen, most important of which is the top one listed, Perfuse Therapeutics, because I'll show you some data from a study that we did under contract with that company. And then also I'll show you lots of images that we obtained using the Heidelberg engineering instrument known as the Spectralis. And that instrument was given to us at no cost to do this research. And so that's important for you to understand. And then finally, I want to thank all the institutions for the support of the work. And although Glaucoma Research Foundation wasn't listed here on the screen because it didn't support the work that I'll show you today, I have received support from the GRF in the past, and it came at an absolutely critical time. So I'm really grateful for that prior support, in addition to the support of the NIH, National Eye Institute, and Bright Focus, and our local foundation here at Legacy. So by way of an outline for today's discussion, I thought I would touch on three different topics or studies that we've been doing recently. You'll see them here listed in the outline. One is topical insulin eye drops for neuroprotection and restoration, specifically in the context of glaucoma. I'll tell you about some of the work that we've done in collaboration with Adriana DiPolo. And then part two will be about the role of blood flow and hemodynamics in glaucoma. And this is where the study that we did with Perfuse comes into play, because they are now in clinical trials with their drug, which aims to improve blood flow. And then finally, in the very last part, I will talk about some of the work that we've been doing in the realm of what's known as repopulation of retinal ganglion cells. So for people whose vision has been damaged and optic nerve has been damaged by glaucoma or other optic neuropathies to a very severe state, is there anything that we could do to restore vision? And the sort of science fiction type phrase we might use is to regrow a new optic nerve. And we start that by trying to transplant retinal ganglion cells into the retina. And I'll show you some results of some of the studies that we've been doing in that regard. Okay, so to start with part one, I mentioned Adriana DiPolo. You'll see her face on the next slide. But over many years now, she had been investigating in a mouse model initially of optic nerve injury that's a surgical and rather traumatic injury, that insulin was serving as a very robust method of regenerating damaged parts of ganglion cells, including the dendrites, which I'll show you in a minute. She went on to then say, well, that's one model of optic nerve injury. What about if we do experimental glaucoma? So she did that study in mice, and I'll show you a little bit of those results just for a bit of context and background. And she also switched from doing injections of insulin into the gut in mice to doing topical eye drops. And lo and behold, it proved to be very, very protective and restored both the health of ganglion cells in their structure and their function, as well as visually guided behaviors in mice. And so this was very, very promising. And with the same grant support, we went on as part of that project to study this in our model here that we conduct in Portland using non-human primates, that is rhesus macaque monkeys. So a larger animal model whose eyes are much, much closer to those of clinical patients, of human beings. Um, as well as the model of glaucoma itself matches the timeline a lot closer than, of clinical glaucoma than does most models that are conducted in mice. So let's look at that. There's a picture of Adriana. And so importantly, the way this study was done is that she introduces into the anterior chamber, you'll see in this picture, hopefully you can see my mouse squiggling around, little beads that block up the outflow of aqueous humor, the fluid that fills the inside of the eye. And with that outflow mechanism blocked a little bit, the pressure starts to build in the eye, known as ocular hypertension, or OHT for short. And the arrows are indicating that pressure inside this little mouse eye has been elevated. And you can see that graphed here in the gray symbols. You can see that pressure was elevated significantly over the course of this study. 3 weeks is shown here. Now, after 2 weeks of ocular hypertension and the incipient glaucoma, her and her team started the topical eye drop treatment. And lo and behold, here's some of the data. Now, I'm not going to take you through all the graphs, but I want you to focus really, if you can, please, on the very top row, because these are tracings, hand-drawn tracings of retinal ganglion cells. And those are the neurons that are damaged by glaucoma. It's their axons. Each of the million or so of the ganglion cells in our human eyes has an axon that collectively leave the eye and become the optic nerve. So all million axons together are what comprise the optic nerve. And that's what's damaging glaucoma. So these are the cells of interest, the RGCs for short, retinal ganglion cells. These cells in a healthy eye will look like this, for example, in this mouse. And then after ocular hypertension or experimental glaucoma in these mice, after 2 weeks of high intraocular pressure, you can see that the arbor, if you think about, let's say, a tree with its branches getting finer and finer and smaller out until the full canopy is reached, you can see that after glaucoma, the arbor is dramatically reduced in its size, its overall area, its complexity, the number of branch points, and so on. Now, starting at 2 weeks and then delivering topical eye drops of insulin or as a scientific control of only saline eye drops, so saltwater eye drops, this experiment showed that the saline didn't really help. In fact, in general, things got a little bit worse 1 week later with ocular hypertension if the mice were only getting saline eye drops. But if they got insulin eye drops, you can see in the top right panel, the complexity was restored in these ganglion cells all the way back to the way it would look in a healthy mouse. That was quite remarkable. And the graphs at the bottom of the page just show you all of the ways of quantifying that, of measuring these things. But really, this is the picture to keep in mind, that retinal ganglion cells were restored with topical eye drops of insulin. And some of the other important things are that the number of contacts of the upstream cells in the retina that are responsible for delivering information that is about the visual world to the ganglion cells, which then deliver that information into the brain. Well, those upstream cell contacts were in much better shape as well in the eyes that got insulin compared to the eyes that only got saline. Okay. All right. So the next step that Adriana and team showed is that not only did this protect the number of ganglion cells in mice, in the eyes of mice overall, that's shown in the lower left, but it also protected their function. And that's what these green pictures are showing here. In a healthy ganglion cell in a mouse that's had genetically encoded in it a reporter, that is a glowing molecule that glows when the cell is excited. And in a normal healthy ganglion cell, that excitation is shown here with the brighter green, and then it goes back to a quiescent state. In the mice that received ocular hypertension for 2 weeks, followed by another week of ocular hypertension with saline eye drops, their recovery, the recovery of this excited phase, was really delayed and prolonged. Whereas the mice that received insulin, the ganglion cells seemed to respond much more like a normal, healthy ganglion cell, and that their recovery was hastened. And the graphs at the bottom just show those measurements done quantitatively. Now, most importantly of all that, because having the right structure of a ganglion cell and having its calcium signaling be normal, those are important steps. But ultimately, what we want to know is, did we preserve vision in any way? And so Adriana DiPolo and her team demonstrated using this rotating bar sort of test that the vision in mice was maintained. Let me explain just a little bit more about that. I'm sure many of you have experienced this when you're riding in a train and you see the scene going by. You can't help but feel your eyes bouncing back and forth as the scene is gliding by. Well, mice have a similar phenomenon. And instead of riding on a train in this case, you put them in a barrel that has black and white painted bars on it, and you rotate the barrel. And the mice can't help, if their normal is vision, but to follow the direction of the rotating bars. And so their eyes will move and their head will move in the direction of the bars. Now, mice that received insulin maintained a normal response to this visually guided task. So the fineness and the contrast of the bars was the same as it would be in healthy mice if the mice with glaucoma got insulin eye drops. But if they got only saline eye drops, you can see between week 2 and week 3 that their vision, their ability to do this task, this visually guided task, declined. So that's where my team comes into play. They're shown here on the screen, my local team, along with Adriana again, where we now said, okay, this worked in mice. Let's see if it works in monkeys. And so we follow very strictly all of the regulatory guidelines that are in place, just for your reassurance. And this slide has a lot of fancy words on it, but let me just say that what we're using here, what this slide describes, is that we're using the same exact kinds of measurements that your clinicians use to detect and monitor glaucoma and any other eye disease for that matter. And they come largely from a technique known as optical coherence tomography, or OCT for short. And so there are a bunch of different measurements. The three main ones here are shown in bright yellow, and that's the thickness of the nerve fiber layer, the thickness of what's known as the ganglion cell complex in the retina, and then the thickness of the health of the optic nerve head in particular. And I'll show you pictures of that so that those words will have some meaning momentarily. Okay, so here is the first, that's the nerve fiber layer thickness. And just this is the kind of thing that is by far the most common measurement that your clinicians will do to detect glaucoma and other optic neuropathies, other eye diseases, and to monitor to decide whether or not the treatment is working pretty well, to maintain the health of the ganglion cell axons. Those axons in the retina are shown here in cross section. Now, this image at the top comes from the location of this green line. And this black and white picture in the inset box here is probably familiar to some of you. It shows you the surface of the back surface of the inner part of the eye. This is the optic nerve as it's leaving the eye. It's called the optic nerve head because it's the very beginning of the optic nerve, like the headwaters of a stream. And in through the same gap in the shell of the eye, known as the sclera, the white part of the eye, that the axons leave, the vasculature comes into the eye. And you can see this artery branching into the eye and its counterpart branching to the inferior part of the retina. And then this blood circulates through the capillaries and into the veins to drain back out of the eye using the same canal in and out of the eye. Okay, so the position of the green line, however, is a scan that when you pull it out and unroll it, you're seeing now in 3 dimensions. So in this black and white image, we would be looking into the screen when we unroll that scan here. And you can see the vessels in cross section, these 2 vessels right here, or these 2 vessels right there. And then these vessels over here is this triplet of vessels down here. And importantly, we want to measure the thickness between this yellow line and this red line, and that's known as retinal nerve fiber layer thickness. So in this particular model of experimental glaucoma, when we started, this eye was healthy, and then it was given ocular hypertension through a laser procedure. The eye pressure goes up over time. I'll show you that in a graph in a moment. And after a period of months, you can see that 20% of the thickness of this tissue has been declined. So that means 20%, actually a little bit more than 20% of the axons in the optic nerve have degenerated and disappeared. And so this is what we're trying to prevent. Another way of measuring the health of the retinal ganglion cells is to scan a different area. I'm going to show you this map. And so instead of that scan being around the optic nerve head, now we're scanning around what's known as the posterior pole of the eye. And this map here is a color map. The warm colors represent the thickest part, and the cooler colors, like in blue, represent thinner parts. This is the healthy state. And then down here in the lower right is that same eye that I showed you in the previous slide at the same final time point where it's lost about 10%. And you can see that the cooler colors are starting to dominate, especially in this superior part of that eye. Okay, and the final of the three measurements that you'll see is known as the minimum rim width, and it represents the thickness of the length of this little green line. And this little green line you can see at the beginning when this eye was healthy, on average at all the different positions around the optic nerve, averages to nearly 300 micrometers. After some damage, you can see what your clinicians have probably told you is cupping, is the word that's used, and that is that the surface of the optic nerve head has been depressed and posteriorly deformed, and the length of this little green line is dramatically reduced. Now that represents in part loss of the axons, the degeneration of the axons, but also the very specific kind of deformations that occur in the optic nerve head in glaucoma, in comparison or in contrast to other optic neuropathies. So anyway, we have our three measurements, and I'll show you those over time. In this model, here are the two eyes. The blue dots show you the intraocular pressure, IOP for short, in the left eye, which is the healthy control eye that we use for comparison. And then after creating aqueous humor outflow problems by laser, you can see that the intraocular pressure goes up. Some of you that have glaucoma will know that an intraocular pressure approaching 30 is not comfortable, for your clinician to monitor. We would all be concerned about risk of progression, but that's actually what we're trying to study here in this model. So intraocular pressure is elevated, and sure enough, those three measurements decline. The red dots going down, the red dots going down, and the red dots going down. I have two other panels on here. Those are sort of bonus, but we'll stay focused on these three, the nerve fiber layer thickness, the ganglion cell complex thickness, and then the optic nerve head rim thickness there. So what I want to show you is three different ways of looking at that. We compare the values when the eye was healthy to the values in this study where half of the animals, once they achieved the onset of glaucomatous damage, they received topical insulin eye drops, whereas the other half of the animals in the study received only saline eye drops. And so we're going to compare the difference between this time point and this time point 16 weeks later to see if insulin was indeed protective in monkeys as it was in mice. We're going to do that in two ways, both comparing those values and then also looking at a trend over time to see if the trend was flatter in the insulin-treated eyes compared to the saline control eyes. I'm just going to show you that the pressure, long and short, it's a busy slide, but the pressure was, the intraocular pressure, IOP for short, was no different in the two groups. So at baseline here, the eyes that eventually got glaucoma started out normal, just like their fellow control eyes. Then after laser, their intraocular pressure was elevated. It was not statistically different in that 16-week treatment period. And that was not different between groups that received insulin in green versus those that received saline in red. Okay, not significant is what NS means here. And that's important because what we want to know is the effect of the insulin, and so it's important that the intraocular pressure was the same in the two groups. Okay, now here are our three outcome measures, the thickness of those measurements that clinicians use to track the status of glaucoma. And so importantly, the first thing to note is that there was no significant decline over the treatment phase in the eyes that got insulin. And that's true of all three of our main outcome measures, plus the bonus one here shown on the right. However, in the eyes that received saline instead of insulin, there was significant progression of all three of these metrics. And so in this scenario, insulin was protective. We've done this analysis in a different way where you take the values of these three different measurements and you compare them to the baseline when the eye was healthy and the same story is true. There was no significant progression in the eyes that received insulin by all three measurements, whereas there was significant decline in the eyes that received only saline. In other words, their experimental glaucoma progressed. Okay, now we do that trend analysis where we measure the slope of that line through the 8 time points over the 16 weeks of the treatment phase. And sure enough, you can see here that the that line was significantly declining. In other words, disease was progressing in the eyes that received saline only compared to the eyes in green here that received insulin. And that was true of all three measurements plus the bonus. If we ask whether that rate of change, that slope of that line was different than zero, in most cases, it was not significantly different than zero in the eyes that received topical insulin eye drops just once a day. Whereas in the eyes that received insulin, it was significantly more negative. In other words, sorry, the eyes that received only saline, they continued to progress. Whereas there was no statistically detectable progression in the eyes that received insulin. Again, confirming that we found very remarkable and substantial evidence of protection against glaucoma progression using topical insulin. Now, we went on because this is laboratory experiments. We can take the eyes and we can study specifically in great detail how the cells inside the eye did. And so here is work done by Priya Chowdhury and Juan Reynaud to measure and count every single ganglion cell in the retina of the eyes that were in this study. And what you can see in this one little area is what we're showing on this one slide. This red box is blown up, magnified here, zoomed in so you can see how many ganglion cells were in the glaucomatous eye that received only saline, and it's a dramatic reduction compared to the fellow control eye. Whereas the eyes that received insulin treatment over that 16 weeks, notably after glaucoma had already started and been underway, have a very comparable number of ganglion cells at this one location. But the truth is, we counted all of the ganglion cells in all of the wedges of these retinas, which is shown on this slide, where we're going about trying to count every single ganglion cell in these tissues. And here is, the bottom right really just tells the story. So here are the control eyes that were the opposite eye of the eye that received glaucoma. Now, those two bars that are in checkers represent, should represent close to the normal number of ganglion cells in the retina. Just to compare that and make sure, we got some eyes from the Oregon National Primate Research Center for which nothing had ever been done to the eyes. So we call them naive. They were naive eyes. And you can see that the fellow control eye number of ganglion cells matches the naive rhesus monkey number of ganglion cells. Remarkably, the eyes that received insulin topical treatment also had the same number of ganglion cells in their retina as all of these control groups, whereas the eyes that received saline continued to progress and lost about half of their retinal ganglion cells over that 16 weeks. Now, when we count the number of axons in the optic nerve, we find a similar pattern, that there was great protection conferred by treatment with topical insulin. They had lost only a small number of their axons from the optic nerve. And again, treatment was started after glaucomatous damage was underway, whereas the eyes that received the control, just only saline, continued to progress and had fewer axons in their optic nerves. Now, just as a matter of safety, we already knew this from other studies that were done for other reasons, but blood glucose was not altered by topical delivery of insulin eye drops. And so that long and short of it, we measured this a bunch of different ways. So it seems like it was very well tolerated. It didn't alter blood glucose because theoretically you could have hypoglycemia or too low of a glucose level in your blood. As people living with diabetes will know, they have to manage their blood glucose levels very carefully. So it looks like insulin was protective in both mice and the non-human primate model. And therefore, it's absolutely warranted that we go on to do clinical trials in people. And the two such trials are already underway. One notably in the Bay Area, where the Glaucoma Research Foundation is located, and that's done by Jeff Goldberg's lab at Stanford University. And they've already demonstrated so far in the first phase of this trial that the topical insulin eye drops are very well tolerated, and they're moving on now to an efficacy phase of the study to see if, in fact, it was protective in the context of clinical glaucoma in human patients, not just in these laboratory studies. There are important caveats to consider. I'll skip over the slide, but one really important one is that I didn't show you this. Our measure of function is from what's known as an ERG for short, or electroretinogram. Much like you can do an EKG to measure the electrical function of the heart, you can do an ERG and measure the electrical function of the retina. We did not have, though, like the spinning bars test or another measure of actual vision in this study. And so it's an important thing to consider. That said, we did show very, robust evidence that topical insulin was a successful neuroprotective strategy. And in combination with the work done by Adriana, uh, where they demonstrated very clearly that it was both restorative and protective, I think there's great promise for topical insulin going forward. Okay, with that, I'm going to shift gears into part two, which is about blood flow and hemodynamics, meaning how blood flow, and very specifically in our case, how it varies during the cardiac pulse. When your heart beats and that rush of blood goes through the eye, we then get to see that in the eye. And it turns out that some of the measurements we do using some relatively newer techniques um, tell us about damage that glaucoma is doing in ways that we didn't really appreciate before this. And I mean, in regards to how the tissue is changing, not so much that ganglion cell, we talk all about so far ganglion cells being injured and their dendritic tree becoming less complex and eventually those cells degenerating and loss of their axons from the eye, but also in the way that the blood vessels are impacted by glaucoma. So I've shown you the way the model goes. We elevate intraocular pressure, and then we make these measurements over time. This circle here is done to show you the very earliest statistical time point that we can detect change has first occurred. So change in the optic nerve head rim width measurement and change in that retinal nerve fiber layer measurement. Again, both of those representing largely the number of axons in the eye, the number of retinal ganglion cell axons in the eye. So at the first detectable time point, this onset of damage time point. I'm just going to show you those slides again here in cross section, just showing you not only the first when the eye was healthy and the final time point, but that along the way, just as the graph showed, you see progressive increasing depth of the optic cup, as it's known. So changes in the optic nerve head that are very specifically indicative of glaucoma damage. But in this study, we're interested in the vasculature. So I'm just going to use, I showed you these slides, so I'll skip over them. They're the same ones just to remind you what we're doing. But now in this study, what we're interested in is how are the blood vessels changing? And so here, I'm going to show you an image of what's known as an OCT angiograph. And that's a way that the OCT uses motion it detects motion inside the eye to make a picture of the blood vessels. Now, the largely, once you stabilize the eye itself moving and everything else, the only thing that's really moving at any reasonably fast pace are the red blood cells flying through your blood vessels. And so that motion makes this very impressive picture known as an OCT angiograph, or OCTA for short. You can see the large arteries here, and then the vein returning back to the optic nerve, as well as all the capillaries in great detail. Now, in this eye, the same eye I showed you from the previous slides, here it is at healthy baseline, and you can probably appreciate at the final time point that there are many fewer of these capillaries, especially here in this location and here in this location, and then down here in this location as well. Here is a map, again, a color map of the thickness of the retinal nerve fiber layer tissue, warm colors being thickest, and you can see that that tissue became thinner at that final time point. So there's a real chicken and egg question here. We're losing the tissue, and we're losing the capillaries within the tissue as glaucoma degeneration ensues. The question is, which comes first? And that's what this study aimed to address. So we looked over time, the same sort of series of measurements. You can see the optic cup getting bigger and bigger as this sort of black hole here gets larger and larger. You can see the thinning of the tissue, and I already pointed out you can see loss of the capillaries. I'm going to show you a graph though, and I'll walk you through it because it's a little bit busy. The blue boxes just represent the control eyes, and they're stable and steady in all six of these plots on the page. So you can more or less ignore those for the rest of the conversation on this slide. Here we already know that the rim tissue, the optic nerve head, gets thinner, and that's shown here. The volume of that tissue is decreasing over time from those earliest detectable time points right there onto the final available time point. We also know that that retinal nerve fiber layer volume gets thinner and thinner, that color map I showed you, and there it is. So that's nothing interesting or new or exciting about that. This is already known. But one thing we did is I'll go back one slide. We separated out the measurements from these bigger vessels compared to where the smaller capillaries are. Now, in the optic nerve head, the amount of large vessel, LV for short, that comprises that tissue didn't change over time, nor did the relative amount of the capillaries change over time in the optic nerve head. But in the retina, while the large vessels didn't decrease, and we don't expect them to until very late stage glaucoma, the microvasculature, the capillaries declined even at the earliest detectable time point of thinning. So we can now say with this paper that was recently published that at least in this model of glaucoma, the vasculature is critically important in that it declines, these capillaries decline at the very same time point that we can first detect any structural change at all. And here's just one example. This technique I mentioned requires motion of the red blood cells to be detected and to be shown in this picture of the capillaries. Now, at this one particular location, if we look over time, you can see here's a capillary in this location that then disappeared on another day and then reappeared on another day. So what happened is this capillary isn't gone, but its function is abnormal such that the flow through there is too sluggish or slow, or perhaps so slow that it's not even existent to be detected by the technique. Now, that's really important because it means that capillary is still there and we could rescue its flow potentially. And that's where some additional work has come in. Oh, I should mention that we're really interested in and published a lot over the last couple of years, also about the pulsatility, so the strength of the pulse going through there. I mentioned that, and I'll show you some videos. This work also recently published, done by Arwa Arashud, who's finishing a postdoctoral fellowship here at Discoveries Insight. I want you, in the next videos I'll show you, to pay attention, please, to the arteries here that are shown by the red arrows in one case and then a different case because they show you the same thing. Now I'm going to play this video and let's watch up here. You can see the pulsation of these arteries, right? You can see that the artery even wiggles in its position a little bit. Now, as it pulses, it also makes these funny little lines here. These little lines we initially called the comb artifact because it looks like teeth of a comb. And they're definitely not little horizontal blood vessels like that. It's an artifact or an abnormality of the technique. But it speaks exactly to the fact that the tissue itself is changing in glaucoma. I should have pointed out this is a glaucomatous eye, albeit at a relatively early state. Now here's another eye that's a little further along in its glaucomatous damage. Let's play the video. You can see the pulsation is very strong, both here at the floor of the optic cup, and then here along this artery, here along this artery, and then these two artery branches down here. And you can see those little lines happening right there. Now, the point of those lines is that they represent changes in the mechanics of the tissue. The tissue is getting stiffer, either or both the wall of the arteries or the retinal tissue around it is getting stiffer and manifesting this abnormality or artifact. So we're really interested in this pulsatility phenomenon because it's proving again and again to be predictive of the rate of progression in glaucoma in human patients when we judge their visual field changes over time, as well as in monkeys in this particular model. So what can we do about that? Well, we contracted with that company I mentioned earlier, Perfuse Therapeutics. I'm going to show you a video of blood flow in the eye. On the left is a monkey with experimental glaucoma before we injected a drug that's aiming to increase blood flow in the optic nerve. You can see the pulse. That's the heartbeat. I'm going to play it one more time. You can see the pulse beating, beating, beating. And what this map is a color map of blood flow or sort of velocity of the underlying red blood cell movement. You can see here in the blue colors that this optic nerve, the head of the optic nerve, has pretty poor blood flow through most of the temporal side. This left side of the image is mostly blue, a little bit of green. When we inject the drug that's aiming to increase blood flow and follow that over 6 hours, I'm going to play this video that was taken almost 6 hours later. There it is. It's mostly back to green and only a little bit of blue. So we've recovered blood flow through the optic nerve head tissue. Let me show you the full sequence or a part of the sequence rather than just videos. Here's that same eye, time averaged now of the whole video to show you the blue showing a defect in perfusion. Capillaries are not having good blood flow. After the drug was introduced into the eye, that blood flow is recovered a great deal. Here's the fellow control eye, which also got the drug, and its blood flow increased a little bit, but the increase was much more in that other eye. Let me show you that in graphical form. We did the study using just a control. So in other words, basically salt water instead of the drug being active. And then three different doses, a low, a medium, and a high dose of this blood flow enhancing drug. And you can see that the glaucoma eyes are in red. The glaucoma eye that I just showed you in that video is in red. And the left eye is shown in blue. Now, again, flow is reduced in the optic nerve head. You can see the red dots are way below the blue dots. But especially with the strongest dose here, by the time we get to the end of the time of measurements, blood flow is recovered nearly to the level of the healthy control eye. The measurements, the graphs on the bottom just show you the measurements normalized to the pre-drug injection time point, just showing again that it's very much related to the strength of the dose. So here's the results of all of the animals in that study instead of just that one example. And it's the exact same pattern. With the strongest, highest dose, by the end of the 6 hour assay, blood flow in the optic nerve head was improved from this start point where it was reduced compared to the fellow control eyes. It improved over time all the way up to be almost about even back here with where the fellow control eyes started. There was a little bit of an increase in the control eyes as well. And that's shown a little more clearly here when you normalize these results compared to their pre-injection time point. That's shown here. There is a little bit of an increase in the control eyes, but the increase in the glaucomatous eyes is really remarkable and dramatic. And so now we're in trials underway. This drug is in clinical trials underway. The first phase has been completed and results are being reported showing that the drug was well tolerated as an implant inside the eye and that it did in fact increase blood flow just as this laboratory experiment showed. So I'm very pleased to say that the work that we contributed to the development of this drug has now resulted in another drug that's in clinical trials with hope for treating glaucoma in a way that's complementary to just lowering IOP. So we might use insulin eye drops or some sort of implant to produce insulin as a restorative for the retinal ganglion cells. And we might use a drug like this one. Doesn't have a fancy name yet. It's just known as PER-001. But we might use this one to enhance blood flow. And the next phase of this trial that's underway now will determine whether or not that, in fact, is protective for glaucoma. Okay. And so we're on to part 3. Well, I told you these conclusions already, but they're written here on the slide. One of the things that we do just before moving on to part 3 is to intentionally manipulate the intraocular pressure. Here's an example of scans that were done where the IOP is set at 10 for 10 minutes, millimeters mercury, that is, a nice normal healthy pressure. And then we increase it every 10 minutes by another 10 millimeters mercury, going 10, 20, 30, 40, 50, up to 60 millimeters mercury, very, very high intraocular pressure. And then we lower it back down to 10. And we do all those measurements I showed you, but I want to show you this video because it shows you how, with increasing intraocular pressure, the surface of the nerve head deforms and the vasculature changes, the reflectance of the axons coming into the optic nerve change. That's shown on the left, the vasculature shown on the right. But you can see all those deformations right there occurring, and we're really interested in how this speaks, how some eyes might, this might inform us about how some eyes are more susceptible to a given level of intraocular pressure than other eyes. Okay, now what do we do for patients whose vision and optic nerve has been damaged to a much more severe state? We're not talking now about early detection mechanisms or, techniques for improving blood flow in order to save ganglion cells, because the ganglion cells in some eyes that are already blind are already degenerated. So is there anything we can do for patients like that? There are a number of avenues that people are pursuing. One of those is known as repopulation, where you can either encourage cells in our own retina to go back to a more embryologic state where they're able to become a different cell type and encourage them to become ganglion cells that are now had been damaged by glaucoma, or in our case, by transplanting donor ganglion cells into the retina. And in this case, the donor ganglion cells are derived from human stem cells. Jason Meyer here, shown on the left, is a stem cell biologist, and he grows retinal ganglion cells in a dish, so to speak. It's a bit like a dish in the laboratory, and then ships them or delivers them by hand to Portland, Oregon, where we transplant them. Here's our local team doing that work. I want to give a big shout out to all of them for the diligence and hard work along the way. But our main question so far that I want to show you some evidence is, will donor cells, these transplanted retinal ganglion cells, survive in the glaucomatous host retina? Will they mature? Will they develop? Will they become integrated? We have a host of other questions, but I just want to show you some evidence that yes, they survive, yes, they mature and develop, and not quite yet at the longest time point we've studied postoperatively have they become fully integrated into the retina, meaning listening to the host retinal cells and their reporting of what the visual world uh, shows. So this imaging work has been led by Kazuhiro Kurakawa, who's an optical engineer with expertise in what's known as adaptive optics imaging. Now, AO for short, adaptive optics, AO imaging allows us to see individual single cells in the retina. And that's where this work comes in. The bright glowing, the cells, I should say, Jason has engineered the cells so that they have a fluorescent reporter. That is, some of you will recall, like a black light poster, and you turn off the regular lights and turn on the black light, and you can see things glowing. Well, that's essentially what we're doing here. These cells are glowing if they're illuminated in just the right way with sort of a bluish green light. Now, the donor cells can be seen here as the bright white against this black background, or in these little locations, much smaller locations than we can image with regular instrumentation like the spectralis instrument I measured. Here in these colored boxes is where Kazuhiro and the rest of us are doing the AO imaging or adaptive optics imaging, much smaller area that we can image, but with much higher resolution, such that you can see all the individual donor cells. And then we can track them over time at the same locations. Here, the location shown in the yellow boxes is from this part of the retina, and the location in the green boxes is shown here. In another eye, you can see that these cells have developed long axons and dendritic arbors that look very much like a nice, mature, healthy retinal ganglion cell. And now we'll follow um, the case that had the longest postoperative time after transplantation. And so you can pick your favorite location, 1, 2, 3, 4, or 5. We're going to go through the 31 weeks of follow-up. I'm just going to page along, and you watch as the cells change their position and then stabilize in their position and now start to grow. For example, in this case up here, or in this case down here, you can see they're growing extensions, or what are known as neurites. You can barely detect that they're there with the regular commercial imaging, the imaging from the spectralis instrument, you can see those bright white dots. But with the adaptive optics or AO imaging, you can really see the details of the cells. And we'll keep going. You can see how remarkable the development of the axon bundles has become. And then, because these are laboratory experiments, we can take those eyes and study them after life. Although the retina is still alive, here Joe Leffler has made these little tracings I'm showing you here to point out that what he sees in his microscope, where he's going to measure the electrical function of these cells, is the same as what we see in life that we imaged using the AO system. Okay, so I'll flip back and forth just to convince you that that's the same cell. And then when Joe puts an electrode into this cell, he can measure that it has absolutely robust, what are known as action potentials or spikes. And that's the way retinal ganglion cells signal what they see, so to speak, into the brain by this pattern of spikes. So this has at 31 weeks postoperatively after the transplant has become a very mature ganglion cell by electrophysiology. Another really important thing, this is his output, the cell's output. On the bottom is the cell's input. So although it's not generating light responses, like if you flash a light onto that cell, and that means that it's not yet quite hooked up into the host retinal cells that are upstream normally from ganglion cells, it does have, it does have the machinery to listen to cells, and that's shown by this trace at the bottom, which are known as spontaneous excitatory responses or postsynaptic potentials. Anyway, excitatory responses demonstrates that this cell has generated the machinery to listen to the host cells upstream. And so that's our next step in this project is to ours, meaning not only our group of laboratories on this project, but the field, the entire field, the working toward repopulation of retinal ganglion cells, has to not only get these cells to survive, as we've demonstrated here, but also to integrate into the host retinal circuitry, and then also to grow an axon into the right place in the brain. And so those two other challenges I mentioned are an outstanding hurdle for the field. But you can imagine someday that kind of work is already well underway, and you can imagine someday the possibility of replacing ganglion cells in human patients in order to restore sight to those that have already been blinded by glaucoma or other optic neuropathies. And so I'll just reiterate that this part three showed that we do in fact see cells surviving out to as long as 31 weeks after transplantation. They migrate, they grow. In other words, they grow neurites or extensions and axons. They exhibit maturation by electrophysiologic measurements. For example, I showed you the spiking behavior that Joe detected with his electrode. And then also they exhibit maturation when we test for different markers in those cells, the kinds of genes that they've turned on, the kinds of proteins that they're making match that of an adult mature ganglion cell. And so we've come a long way in this particular field that 15 years ago or so I was thinking was pretty much science fiction. And so it's a lot less fictional now, and we have at least the groundwork or the basis to continue on with that work. And so with that, I thank you and look forward to answering any questions you might have in our discussion.
Brad, that was amazing. Really. Thank you so much.
Well, I'm
And
glad. I hope the rest of the audience thought so too.
and we definitely have questions to get to. I think my first very facetious question is, where can I purchase some of those images? Because they look like artwork. Like
Oh, right.
I need some of that in my office.
Well, if you're talking about these blue ones, I'll ask Kazuhiro to send you some, or
Perfect.
I'll ask his permission for me to send you some. Yeah.
Okay, perfect. Thank you. Thank you. Okay, so you presented us with some really fantastic, these three wonderful potential treatments between insulin and what a better understanding of blood flow could do, and then, of course, these stem cells. Now, patients on this webinar want to know at what point, when can they expect some of these treatments to be available?
Yeah. So there was this wasn't completely random in terms of the order. So insulin is already used to treat problematic what are known as so corneal problems, I'll say more generically. So neurotrophic keratitis is the technical term, and insulin is already in use for treating for helping essentially like a degenerative neuropathy in the cornea. So now we know that it may also help uh, glaucoma, and those trials are underway. So I think the timeline for topical insulin is sort of very soon. Like within the next few years, I imagine these trials will be done and we'll see if it's really working in where it really matters in people. And
Mhm.
then the blood flow drug I mentioned is already also in clinical trials. The first phase, just like the insulin one, the first phase has been completed, and that was safety and tolerance. And it was proven very safe and well tolerated. So the next phase of that study is to see will it actually help in the protection of against glaucomatous damage. And so again, that trial is just now getting underway. We anticipate that would take a couple of years to complete. So I think the timeline for both of those is pretty soon. Now, the stem cell thing, that's a very different timeline, and that's why it came last. That's going to be, I'm going to guess decades, but a lot of this depends on how different funding agencies see as a priority this versus so many other things that need to be done. And, how many groups are solving problems along the way and collaborating nicely, et cetera. Maybe it will be a little faster than a couple of decades. I would hope so.
Yeah, I hope so too. It looks really exciting. On the topic of clinical trials, how can patients find out about ongoing clinical trials and learn whether or not they qualify?
Oh man, that is a fantastic question. A little outside my immediate realm or expertise. I think the GRF is in a great position to inform patients about what's available by way of trials for getting involved in a trial. I know for sure that Stanford University is conducting one such trial for topical insulin. I imagine, I'm guessing because they also have the blood flow instrument. I didn't mention its specific name. It's called laser speckle flowgraphy, or LSFG for short. It's People are referring to it as a relatively novel technique. We've been publishing on it for 15 years already, so I don't see it as terribly novel, but it's used very widely in Japan, not as much in the United States. I think only maybe 3 or 4 institutions in the US have an LSFG device. Stanford happens to be one of them, so I imagine that they'll be one of the sites doing the blood flow trial as well.
Okay,
I know your
great.
audience might be from far and wide around North America, maybe beyond for that matter, but maybe
Mhm.
also, consist of a lot of people in the Bay Area. So I would keep an ear out, so to speak, for what's happening at Stanford or UCSF or UC Berkeley, et cetera. Some of the bigger institutions, in other words.
Great. And I do just have to say that GRF does have a clinical trial finder. It's part of our patient resources section on our website. So please, if you're a patient, poke around the patient resources section and you can find it. Okay, so we got a couple questions about the topical insulin. I guess the first question is how is the insulin working? Like do you think it's conferring neuroprotection? Is it going to the target tissue? Is it upregulating like a second messenger? How is it
That's right.
getting to the
Oh, how
brain?
much detail to go into here. So, this is exactly Adriana's expertise.
Hmm.
Adriana, Dr. DiPolo, whose picture I showed and whose work, uh, whose initial work in this area has led to the collaborative project that we did. Um, so Adriana has worked out in a lot of detail, and her team in a lot of detail, the mechanism by which insulin, at least one mechanism by which this seems to be working, because when you block that mechanism, it doesn't work. It works independently of intraocular pressure, and it does, you mentioned exactly, it does work through what you might call a second messenger inside the cell. And so there's a very specific protein that needs to be phosphorylated at a very specific residue along that protein for this action to be efficacious. There are sort of two branches to it. One branch, the molecule is known as mTOR for short, or the mammalian targeted rapamycin. I think it's probably a little bit beyond the scope of our discussion today, but there are two branches of action of that molecule, and One of them is seems to be responsible for the growth of the dendritic arbor, so how widespread those dendrites are, and the other for their complexity or their branching pattern. And so the combination of the two of them sort of confers the full restorative potential of insulin actions. Um, and I should say that Adriana has gone on to do a bit more work to show that there may be some other mechanisms by which insulin is also contributing to the restoration of health inside the ganglion cell.
Great, great. Um, and that work is published, yes, so that
Yeah, the
someone could go
one
look it
paper
up.
that I showed is in Science Advances, uh,
Okay.
and I'm going to say that was the second half of last year. I, I
All
could
right,
I
great.
could look real
So
quick for that.
hot off the presses too. Excellent.
Yeah, relatively. While you're asking
Um,
the next question, I'm going to page through my own slides. I'm no longer sharing, is that correct?
correct.
Okay. Yeah, so it was October of 2024 that paper came out.
Oh, okay, great. And how was the concentration and dosing regimen of topical insulin determined?
That is another great question. We looked through the literature. There was some work done a long time ago by an investigator named Jimmy Bartlett at University of Alabama, Birmingham. Now, at the time in the 80s, he was interested in whether or not you could use eye drops of insulin to control your blood sugar instead of having to take injections of insulin, right?
Mhm.
Subcutaneous, injections with a needle, which, most people probably would prefer not to do. And maybe an eye drop is a nuisance, but less of a nuisance than injecting with a needle. So he did that work at the time and found that insulin was very safe and well tolerated, but it didn't do that therapeutic effect. So you couldn't use it to treat your diabetes. But we knew what doses were going to be safe and well tolerated, and we started with that. Um,
Okay.
and I had a slide that showed it was 100 units a day, and that's the study now that's being done at Stanford is comparing one such drop per day, as we did in monkeys, versus two or even three drops per day. And so far, the initial phase with as many as two drops per day showed that it was very safe, well tolerated, no side effects, etc., except for stinging, upon putting the drop in the eye, etc. But minor things that most eye drops do. Um,
Yeah,
and now the efficacy trial, whether it's actually going to be protective against visual field progression, et cetera, is being done with one, two, or even three drops a day. There are different groups, to my understanding.
great. You brought up a good point as you were talking about the topical insulin that you gave the model 2 weeks to develop before you initiated treatment. And so that it gives the disease a chance to develop before you actually see if it's going to be protective or restorative. So, what do you think that means for patients with late stage glaucoma? Could some of these treatments be potentially beneficial for them as well as people who have early stage?
Right. Um, Great question. A little bit different in mice than we did in monkeys. The mice, they their glaucoma, so to speak, uh, progresses very rapidly. So 2 weeks is
Mhm.
enough to already start to see the very beginnings of damage, as the slides showed. In monkeys, we waited until we had those measurements showing damage was already underway through
Mhm.
OCT. Rather than a specific timeline, we actually just made because we wanted it to be translationally relevant. Patients don't know when exactly they're going to have glaucoma. It gets diagnosed after it's already occurred. So we wanted this to be relevant in that regard. Now, going down the road, as you point out, I think as long as you have some ganglion cells in your eye, it stands to reason that insulin should help and be restorative in that regard. That said, I'm a scientist. I hate to ever speak without evidence. So we don't know exactly from the point of view of evidence just yet if that's true, but it should be true. It stands to reason. And so we'll have to see how these trials shake out.
Great. Um, as an imaging specialist, a question that came up is how reliable are current tests like visual field testing and OCT for monitoring glaucoma?
Right.
And are there like advancements coming down the pipeline that we can look forward to?
Absolutely. So just statistically speaking, the OCT measurements are more reliable in a very specific definition of that word reliable, meaning if you do the test, you know, twice in a day or twice within a week when you know that nothing's actually really changed inside the eye, the two measurements will come out much closer together than if you do the visual field test twice on the same day or twice within a week or something like that. It's much more variable. Another big problem for glaucoma that's very well understood is that just in the range of where glaucoma is really getting quite significant and impacting vision, in other words, visual field damage has become a little bit more substantial, is when the test is least reliable. When you're totally blind, for example, God forbid, um, the test is very reliably going to say that. When you have very, very good vision, the test is very reliable at saying, yeah, the vision's pretty good. Where it becomes most unreliable is where we need it most. Yet it's the measure of function. It's this absolute standard of measure of how the visual world is being perceived. And so anatomy is one thing and measurement of structure by OCT is one thing and very, very important. But in the end, we want to know how our vision is doing, not just the cells that produce our vision. And so, alas, I think we need them both still, even though one is distinctly more reliable than the other.
Okay. Well, I think we have to finish the questions there, but we're so thankful for your time and your expertise today, Dr. Fortune, and thank all of you for taking the time to be with us today. And thank you for Brad for providing such helpful information for glaucoma patients
My
and
pleasure.
family
I'm
members and
just sorry
friends.
I didn't leave more time for questions. That's an important.
It's okay. It was fascinating work. Thank you once again for being here today and for standing with us in our bold vision of a future free from glaucoma. Because of your unwavering support, the cure truly is in sight.